Hi everyone. So the topic for this week's class is microbial genetics and genetic engineering. What is genetics? It is a science of heredity, something that is passed from one generation to another generation. So this also includes the study of what our genes are, how they carry the information, how the informations are being replicated and passed to subsequent generations or between organisms, and how expressions determines a particular characteristics. So basically, in these genetics fields, you are actually studying about the genome or the DNA. The whole of the genetic informations in a cells are actually stored in a structure called the genome, and the genome contain either chromosome and plasmids or chromosome only. The chromosomes are actually the structures that contain DNA that carry the information. So when we talk about DNA, you should imagine that DNA contain or composed of the 4 nucleotides, all right, A, T, C, G. And those informations in the chromosomes are actually in the form of genes. Genes are the segment of DNA that code for functional products. So what are the functional products? So you will learn about this in the next few slides. Where can you find the genome of the cells? So basically, these are the general locations and the forms of the genome, and in two cell types and selected viruses. So we have eukaryotic cells. The chromosome or the genomes are actually the chromosomes and plasmids. The chromosomes are located in the nucleus, all right? And then whereby the plasmids are roaming around freely in the cytoplasm. Whereas in the prokaryote, so prokaryote doesn't have a nucleus, right? And then so the chromosomes are and the plasmids are in the cytoplasmic cytoplasm. The viruses contain either DNA and RNA. So yeah. This is a picture of actually an E. coli chromosome. So the E. coli has basically around 4.6 million base pair of DNA, the ATP, the nucleotides, and it is about 1 millimeter long. So the size of the size of E. coli cells is around in micrometer, right? So that is 10,000 longer than the entire cells. So the structure of DNA is actually supercoiled. So it is actually looped and folded and so on. So because of this supercoil, then the size of the chromosome can be shrinken or can be shrunken to about only 10% of the cell's volume. DNA is a short form for deoxyribonucleic acid. It is a long chain of nucleotides which consists of the deoxyribose, a pentose sugar, a 5-carbon sugar right here, and then it has the nitrogenous bases, which either this is the basis, so which either purine or pyrimidine. And also DNA contain a phosphate group, phosphate here. The nitrogenous base of DNA is either pyrimidine or purine. So you can have a look at the structure here. The pyrimidine has 6 ring carbon ring molecule, whereby purine has the 6 and the 5 structure, 5 carbon structure here. And because of this nitrogenous means it's contained the nitrogen in the structure. So for pyrimidine, they are either cytosine thymine, or uracil. So thymine here only present in the DNA, the structure of DNA, whereby uracil is only present in the RNA. For purine, they are either adenine or guanine. Okay. So now you know DNA is a long chain of nucleotides, and nucleotide is composed of a nitrogenous base, sugar, and a phosphate. So those nucleotides in DNA, those nucleotides are arranged in the two long strands that form a spiral called a double helix. Okay, so this is the double helix. So each helix has a series of nucleotides. So we have nucleotides here, and a nucleotide here, and those nucleotides are actually held together with a phosphodiester bonds between the hydroxyl group of the adjacent sugar residues in the nucleotides. And the helices themselves are actually held together between these helices. So they are held together by a bond called hydrogen bond between the nitrogenous base G, where G will pair with C, and air and adenine will pair with thymine. To have a closer look at the bond that I've mentioned before, so this is a double-stranded DNA. This is actually a nucleotide, right? A nucleotide, and then you have between the sugar, you have the hydroxyl group, and then they're held together by phosphodiester bonds. The hydroxyl and the phosphate, whereby in between the two strands of DNA, you have a pairing of the nucleotide, right? So at the nitrogenous base, you have T pair with A, G pair with C, all right? And for the purine, T and A, you have you have hydrogen bond with two hydrogen bonds, whereby between the pyrimidine, the G and C, you have three hydrogen bonds. Please take note that the two DNA strands are actually paired in the opposite directions. For paired bases to be next to each other, the sugar components is upside down relative to each other. The end of the hydroxyl attached to the 3 carbon is called the 3' end, whereby the phosphate attached to the 5 carbon is called the 5' end. So this is called an anti-parallel arrangement. So one helix runs from the 5 to 3 5 prime to 3 prime direction, whereby the opposite strand will run from 3 prime end to the 5 prime end. Other than DNA, there is also another form of genetic materials called RNA. So DNA is the deoxyribonucleic acid. RNA is the ribonucleic acid, right? So based from the word, the term deoxy means the lack of oxygen. So here you can see there is a lack of oxygen in this structure, whereby in the RNA has oxygen, so form hydroxyl OH here, right? So these are actually this is the only difference between DNA and RNA. The rest of the structure is the same.